Have you ever stopped to ponder how the world would appear if the vibrancy of a sunset or the lush hues of a forest were muted? Imagine seeing the world in a limited palette, where reds and greens become indistinguishable. This is the reality for those living with color blindness, a fascinating phenomenon that alters the way individuals perceive the world around them. In the realm of the human eye, 3 types of color-respective cones exist, each responsible for detecting different color wavelengths. These cones allow us to perceive the vast spectrum of colors, from the deepest blues to the most fiery reds. But what happens when one of these vital cones is missing? When a person lacks a specific group of cones, their color perception shifts dramatically. For example, the green, yellow, orange and red colors that fall between the wavelengths of 525 and 675 nanometers are typically distinguished by the red and green cones. If either of these cones is absent, the person can no longer distinguish between these colors, particularly red and green. This condition is known as red-green color blindness. Red-green color blindness is a genetic disorder that predominantly affects males. The genes encoded in the female X chromosome are responsible for creating these cones. In females, color blindness is extremely rare, as at least one of the 2 X chromosomes typically carries the normal gene for each cone type. However, males possessing only 1 X chromosome can experience color blindness if the gene is missing. The X chromosome in males is always inherited from the mother, making color blindness a condition passed from mother to son. Approximately 8% of women are carriers of color blindness. The effects of color blindness vary depending on which cones are absent. A person lacking red cones, known as a protanope, experiences a noticeably shortened visual spectrum due to the absence of red cones. Conversely, a person missing green cones, or a deuteranope, maintains a normal visual spectral width due to the presence of red cones. However, a deuteranope can only distinguish between 2 or 3 different hues, while a person with normal vision can perceive 6 unique hues. To detect red-green color deficiencies, the Ishihara test is commonly used. Named after its creator, Shinobu Ishihara, a professor at the University of Tokyo, this test was first published in 1917. In this test, viewers with normal color vision should clearly see the number 74. However, those with red-green color blindness may read it as 21. To conclude, color perception is a fascinating and intricate aspect of human biology. It's a phenomenon that shapes our understanding and experience of the world around us. From the genetic factors that contribute to color blindness to the different ways it can affect color perception, the science behind color blindness is a testament to the complexity and wonder of the human body. So the next time you marvel at a vibrant rainbow or a picturesque landscape, remember the intricate network of cones and genes that allows you to appreciate such beauty.